My dear student, colleagues, and all the viewers who are watching this program live from Facebook page and YouTube channel, I'd like to welcome you all to our International Physics webinar. Good evening to all here in Bangladesh, and a very good morning to all those who are watching this program live from USA. So hope you are well and safe from Corona pandemic. So as you know that we are staying in a Corona pandemic situation, so it's very difficult for us to continue our normal academic program inside the campus. So we have to start our online program. I think you have already come to know that the Department of Physics, Pabna University of Science and Technology has started its online international physics webinar, including online international physics webinar. So uh, we have successfully completed our 186 international physics webinar. Today it's our 187th international physics webinar. So we are trying to adjust with this new normal situation. So today it's very important day for our department, and today I would like to welcome you all to a joint session between our department, Department of Physics, Pabna University of Science and Technology, and the Department of Physics and Astronomy, Department of Molecular Biosciences, and Northwestern University, USA. And we have with us here today Dr. John F. Marko, sir, Professor, Department of Physics and Astronomy, Department of Molecular Bioscience, Northwestern University, USA. And he has already connected with us, so I'd like to welcome our speaker, sir. Good evening and good morning to your parts, and thanks for accepting our invitation, sir. So, and we would like to say thanks on the behalf of the Department of Physics, Pabna University of Science and Technology, for accepting our invitations. So before going to you, I'd like to introduce you with our student. So for those who are new, I'd like to inform you all that we have divided our webinar into three parts. First of all, we'd like to introduce our speaker with all of you, and then our speaker will deliver his speech, and at the end we have a discussion session. In that time, anybody can join with us, and you can ask question by commenting also. I think you have already come to know the title of this today's international physics webinar. Title is the loop extraction and chromatin cross-linking and geometry, topology, and mechanics of chromosomes and nuclei. And our speaker is John F. Marko, Professor, Department of Physics and Astronomy, Department of Molecular Bioscience, Northwestern University, USA. And you can see his short bio. So Professor Marko works on an application of statistical mechanics and polymer physics to biophysical problems, and in particular to the micromechanical studies of the DNA protein interaction, and the chromosome structure. At present, Professor Marko's research group carries out both experimental and theoretical research, and he did his PhD from the MIT 1989. And you can see his educational and professional background. So he completed his bachelor degree in physics from University of Alberta, Canada, and PhD from MIT 1989, and postdoctoral work at University of Chicago, and as a fellow Rockefeller University in biophysics, and joined as an assistant professor at the University of Illinois, and he stayed there up to professor in 2006, and from 2006 to present, he is working as a professor of the University of Northwestern, USA, and this is awards and honor: National Science Career Award 1998, University Scholar Award, University of Illinois 1999, American Physical Society Fellow, Division of Biological Physics 2006, Catalyst Award, Chicago Biomedical Consortium 2007-2013, and research interest. Their lab use biophysical methods, so with particular emphasis on the micro manipulation of single DNA molecules and single chromosomes to study the internal structure of the chromosome in vivo, and to study chromosome organizing proteins and the DNA topomerases in vitro. They have also developed mathematical models relevant to these types of experiment. Projects in progress involve combining fluorescence microscopy and force microscopy in experiments on DNA protein complexes and the whole chromosomes, and in vivo studies of coupling of the chromosome dynamics to gene expression. And this is his Google Scholar ID. You can see his citation is more than 19,000, and this is just selected publication list. So thanks all of your patience. It's time to go to our speaker, sir. I'd like to welcome you again. It's your time, sir. You can start your session, sir.
All right.
And thanks for joining with us.
My pleasure, and it's honor
Yeah,
to speak at such
yeah,
at a distinguished university so far away. It would be really fun to
thank
visit sometime, but it's great to
you. Yes,
present
of course. I
in
will
Bangladesh.
definitely invite you.
Oh,
Yeah.
well, it'd be great. So our group is applying ideas and methods really from soft condensed matter physics and polymer physics to the problem of understanding how chromosomes are organized inside living cells. And so many of you know that chromosomes are made of DNA molecules and they're terrifically long and can easily get entangled with each other inside the nucleus of a cell. And in fact, it's long been thought that during cell division, chromosomes are kind of condensed together, stuck together. But this is a bad idea because it will just make them more entangled. So I'm going to tell you a little bit about how chromosomes are actually organized. They're organized into loops, which are generated by motor enzymes, which burn energy to organize chromosomes actively into loops that are going to end up to be non-entangled with each other. And those motors are called SMC complexes. I'm going to tell you a little bit about them as well. And then I'm going to tell you about chromosomes in the cell nucleus that are where you do have regions of the chromosomes which are stuck to themselves under certain circumstances. And I'll tell you a little bit about how that's playing a role in organizing and protecting the cell nucleus. Okay, so this is a movie of a cell which has a big fat nucleus here. Let's see, where's the laser pointer? There it is. There's the big fat nucleus of this cell. This happens to be an amphibian species of cell, and this nucleus, this round thing, is about 50 microns across, particularly large. And the chromosomes in it are huge. And they've been replicated at the end of the cell cycle. And now you're starting to see them appear as individualized chromosomes. Each one of those chromosomes for this species of animal contains about 3 meters of DNA and or on the order of a meter of DNA, the whole human genome in each chromosome. And you can see they're isolated strings. And the cell moves them around inside the cell after they fold up into this sausage-shaped organization. And then the two copies of each chromosome, which are kind of joined together along the length of the chromosome, are pulled to opposite sides of the cell. So this is a mechanical process, statistical mechanical process, and it's a very interesting pattern formation process, self-organization process, polymer organization process, and it involves space and time, but also forces. We need to understand how these machines work, how the machinery works that coordinates all of this by not just understanding space and time, but also the forces that are involved. And so biophysics and biological physics have been important tools in understanding cell biology, especially in the last 10 to 20 years. Okay, so inside even simple organisms like a bacterium, a bacterial cell, for example, E. coli, which is every one of us has lots of E. coli inside our digestive system, which is helping us digest our food. Each one of those E. coli cells is just something like 2 or 3 microns in length. But inside the chromosome is a piece of DNA that's 1,500 microns. That's 1.5 millimeters long, or about 4.5 million base, about 4.6 million base pairs long. And in fact, when E. coli is happily growing inside you and rapidly dividing about once every half hour or so, there are multiple copies of the chromosome, about 2.5 to 3 copies of the chromosome inside the cell at any time. So there's a tremendous length of DNA packed into those tiny little cells. In a human cell or in a newt cell, I showed you a movie of a type of a species of newt or salamander. In that newt cell, there are 22 DNA molecules, 22 chromosomes. Each one is about a meter in length, and those are all packed into a nucleus, which is about 50 microns across. So the cell has to take some care to organize those DNA molecules so they don't get hopelessly entangled with each other and just turn into a big polymer mess. So because the bending of DNA occurs on such a small length scale. On about 50 microns, a DNA molecule can be bent by thermal fluctuations. This is really a problem of flexible polymers. And so we've been applying a lot of ideas from polymer physics to understanding how chromosomes are organized inside the cell. So in fact, polymer physics is the sort of now viewed as a kind of fundamental starting point for understanding chromosome organization. So I have to present to you a little bit of some of the important components that I'm going to be talking about, the molecular components which do all of this self-organization. So over here is a DNA molecule. DNA molecule is about 2 nanometers thick and very long. And in our cells, it's bound to a bunch of proteins which form complexes of very well-defined structure, about 10 nanometers across. Those are called nucleosomes. So actually, our own chromosomes are polymers not of exactly of DNA, but more like polymers of these little particles called nucleosomes, which are about 10 nanometers across and which organize into thicker fibers. Now, inside the cell, there's a ton of different types of proteins that organize DNA. In fact, a good fraction of all of the proteins that are encoded in the genomes of cells actually are involved with interactions with DNA. And so some of these bend DNA. There's a protein called HMG-B1, which bends DNA, and there are many proteins which bend DNA. Some of these cross-link DNA together. There's something called BAF1, which plays an important role in sticking DNA molecules together. Other molecules play an important role in organizing the process of gene transcription, the expression of genes. CTCF is a good example of a transcription factor, which is also involved in the organization of chromosomes. But I'll talk briefly later in the talk about HP1 alpha, which is a very prevalent protein inside human cells and other types of eukaryote cells, which is involved in cross-linking and sticking DNA to itself. So I'll talk about that near the end of the talk. But right now I want to talk about a protein called Topo2, which I'm going to mention a few times. I'm not going to talk a lot about it, but it's very important because this is an enzyme which binds to 2 pieces of DNA and then cuts 1 piece of DNA and passes a second piece of DNA through and then sticks the 2 DNA molecules together. That involves cutting 2 covalent bonds of the double helix, click, and then passing a DNA double helix through that hole and then sealing them back up. And that's all done. Actually, that takes a little bit of stored energy to accomplish in the form of ATP inside the cell. That's all done by one enzyme. And so if you think about it, that allows, Topo2 is the enzyme that allows disentanglement to occur by cutting one DNA, passing another DNA through. And therefore, you can unknot any kind of knot. The people that are knot theorists, people that know a little bit about knot topology in the room will know that as long as you can pass a string through another string, you can untie any knot, and it's sort of obvious that you can do that. All right, but Topo2 by itself can't tell whether a particular passage of DNA through another DNA is going to add knotting or take away knotting. So you need some other enzymes to tell Topo2 what to do. And it's becoming increasingly obvious that so-called SMC complexes, which are very large, all of the molecules on the screen here are shown at about the same scale. So the DNA is 2 nanometers thick. These nucleosomes are 10 nanometers across. Topo2 is a little more than 10 nanometers across. These guys, the SMC complexes, are 50 nanometers across. That's huge for a protein complex. And in fact, they're organized into rings of 3 proteins. You see SMC2 in condensin, SMC4 in condensin, and then a third protein makes a kind of a ring, and DNA can go through this ring. Okay, so that we believe is important for the functionality of these complexes. So these complexes are very important in organizing DNA, and in particular, telling Topo2 how to remove entanglements. And I'm going to describe that to you in a little detail in the next several slides. Okay, so I'm going to tell you a little bit about the cell cycle as well. Our own cells grow by starting in a phase where the chromosomes are spread out a little bit inside the cell and genes are being expressed, proteins are being made, the cell can grow, and eventually it grows to be big enough that it starts to replicate its DNA. It says, okay, time to divide, and replicates its DNA, and then the DNA organizes into these long visible strings that eventually become visible at metaphase. And so I'm going to tell you a little bit about metaphase chromosomes and experiments we've done on them, on their basic physical properties. Then the chromosomes are separated and they form new nuclei, and then the process proceeds. So our own cells, as you probably know, divide by binary cell division, replication of DNA, separation of the two copies of the DNA, formation of a new nucleus, and that cycle continues throughout your whole life. So the metaphase chromosomes, we know, have a kind of loop organization where you can see in the electron microscope that the DNA and nucleosomes, you're actually looking at the nucleosomes here, little kind of dots that you can see along these fibers, are organized into what looks like loops. And this is actually electron microscopy picture from Ulrich Lemley's group from several years ago, showing this kind of loop organization. Now, the question is, how are these loops folded up? What kind of machinery can do this without seeing the whole structure? And as far as we know, there is no sequence programming of this structure. This is a kind of spontaneously organized pattern for the cell. So it's really a problem in condensed matter, soft condensed matter physics. How do these metaphase chromosomes get formed? And you can see that there's two copies, which are the two replicated copies of the DNA that are joined together at a region here. Some of you may know that's called this centromere region, and I'm not going to talk too much about it, but at metaphase, the two chromosomes are actually two long polymers organized into these brushy structures that are attached to each other. Okay, so we've done a lot of experiments on chromosomes using methodology, which basically is surgery on individual cells. We use small glass pipettes, which have a tip in the micron range of size, and here's a picture of a pipette, and in this particular experiment, the width of this tip is a couple of microns in size, and we can suck a chromosome into that pipette and it will stick to it. We can attach it to another pipette. We can adjust the stiffness of these pipettes so that they can be used as force sensors in the piconewton to nanonewton range. And that's the range of forces relevant to cells and molecules. Molecules, 10 to the minus 12 newtons, piconewtons. Whole cells, about nanonewtons, 10 to the minus 9 newtons. Molecules around piconewtons, cells around nanonewtons. And then we can carry out a mechanical experiment and vary things inside the cell and look at how the mechanics change. And so, in fact, for the newt chromosomes, as we stretch out a chromosome that's originally 16 microns long and make it 50% longer, you can see that it's a few hundred nanonewtons to do that. And there's nice elasticity of these metaphase chromosomes. I'll show you how it looks when we're taking a chromosome out of the cell. This movie was made many years ago by my former student, Michael Poyer. And what you can see is that the cell will open. I'm just going to jump ahead a little bit. Movie. If I can. Let's see if it will. I'll be so happy to let me do that. But I'm going to let you see how Mike is opening the cell. And the chromosomes come out of the cell, and you can see they're flexible and soft. And then Mike is able to pick up, and the current people that do this type of experiment are able to pick up those individual chromosomes that are like kind of soft sausages. And these are tremendously long polymers that are folded up in this loop organized fashion. Let's see if I think I can jump ahead. Oh, yeah. Maybe if I pause. All right. Well, anyway, there's an isolated chromosome, and then Mike is bringing in a second pipette, and he's going to grab that end of the chromosome right there with a second pipette, and you'll see he's able to suck that into the pipette with a little bit of suction, just a few pascals of suction, and then the chromosome will be really stuck to the pipette, and then it can be manipulated and we can do mechanical experiments. There's Mike coming up to the chromosome. And then it's attached to the pipette. And you'll see that as he manipulates it away from the rest of the genome, it's attached by remnant fibers of DNA to other chromosomes. And so that fiber needs to be broken. And he'll do that by attaching another pipette to the other end of the chromosome and then manipulating it away from the rest of the cell. So you get the idea of how these experiments work. Now, for the sake of time, you can watch that movie. It's on our website if you want in its entirety. But so how this works is we open a cell, the genome comes out, we find a chromosome, we attach it to a pipette, and then we attach the other end of the chromosome to another pipette, and we end up with an isolated chromosome that we can do experiments on and do mechanical studies. Okay, so the basic story of individual metaphase chromosomes is they're nice elastic objects. So there's one pipette moving, the left pipette is moving and is causing the right pipette to bend. And we can calibrate that bending stiffness and determine from that what the force versus extension properties are of this chromosome. This chromosome here is from a newt, and it's about 20 microns long, and it's being stretched by about being stretched by about 50% at an added length of about 10 microns. So there's what a newt chromosome looks like. They're large, and a human chromosome. So actually, for many years, we've really been working strictly in human cells. And so there's a chromosome from a human HeLa cell, very commonly studied cell line. And what you can see is that although the human chromosome is a lot smaller and requires much less force to stretch out, it's more on the tens of piconewtons scale instead of the hundreds of piconewtons, this is fractions of a nanonewton or thousands of piconewtons. They both have this nice linear stretch release character. And in fact, both chromosomes, the newt and the human, both have an effective Young's modulus, which is about the same, around 400 or 500 pascals. So if you want to think about the Young's modulus of typical materials that are maybe solid plastic, maybe you're sitting on a plastic chair, the Young's modulus of that chair would be in the 100 million Pascal range. So here we're talking about an object which is only a few hundred pascals. And so that's a very soft material. It's actually rather typical kind of modulus for a very soft polymer gel. But nevertheless, these are very well-behaved elastic objects when you just take them out of the cell. And they're full of solvent. Actually, we can use treatments of a chromosome. Here is a chromosome that's being sprayed with positively charged multivalent ions. Now, the DNA and the nucleosomes have a net charge with the surface charge, which is negative. So if you bring in a positively charged multivalent ion with, say, +2 or +3, that will stick to a negative charge and leave a positive charge to attract another negative charge. So positive multivalent ions tend to act to collapse chromosomes. And in fact, this chromosome volume goes down by about 60 or 70% when this particular polyvalent positively charged ion is used, meaning that most of the volume of the chromosome is actually small molecules that can be displaced. So these are not polymer rubbers, but more like polymer gels where there's lots of mobile, we could call it solvent. It's water and small molecules that can be easily displaced. Now, those originally nice elastic chromosomes, we have a lot of tools in molecular biology for cutting specific types of molecules. And in fact, in this experiment, which is an old experiment that Mike Poirier did again, we expected to cut away the loops and to leave the interior skeleton of the chromosome. But to our surprise, when we cut DNA only, we're not cutting any protein. When we cut DNA, the whole chromosome dissolved. And in fact, eventually, the chromosome turns into kind of a droplet that will evaporate. So it's only DNA that's holding the whole chromosome together. So we start to see a picture that really what the chromosome is like a piece of gel, a loop gel with a lot of solvent inside it. And if you cut up the DNA, you don't have to do anything to the protein. If you cut up the DNA, the whole thing falls apart. So the proteins are there, very important for organizing the chromosome, but all of the structure comes, all of the connectivity and all of the mechanics comes from the DNA itself. So in addition to being the genetic code, carrying the genetic code, the DNA in the chromosome is providing the mechanical stiffness. So our picture is that there's some kind of loop structure to the chromosome, and then some kind of linkers. We know that they're proteins from various other areas of information, and they're most likely the SMC complex. And so we could ask the question: Are the condensin SMCs the main cross-linkers inside the mitotic chromosome? And so indeed, if we label the SMC complexes, we can see that they're distributed along the length of the chromosome. And if we stretch the chromosome out, you can see that there are these clusters of SMC, of condensin SMC complexes. And so indeed, those proteins are playing the role of some kind of like local organizers of the chromosome. So, and here's a similar kind of imaging, actually using a different labeling technique to show you that you can really see these little clusters of SMC complexes along the length of the chromosome. Well, so I want to introduce now what we can do when we do differential studies of cells that have been treated in different ways. So, for example, we can use a method which is called small interfering RNA molecules. RNA has been in the news a lot because of the COVID vaccine, very successful messenger RNA-based vaccine. But before that, RNA was a powerful tool used in all kinds of other ways as well. And it's possible to inject RNA molecules into cells and to block the production of particular proteins. In fact, this is a method of interfering with particular proteins. And so what we've done here is we've taken our HeLa cells and then injected them with these different cells down here that have been injected or actually transfected with small RNA molecules, which then block the production of SMC2. And what happens is you don't see any more SMC2 as imaged here using antibodies inside the cell. And what you see is that the original chromosomes, which come out as these nice sausagey, well-isolated chromosomes, 24 hours after exposure to the RNA, which interferes with condensin, you see kind of blobby chromosomes. 48 hours, very indistinct, disorganized chromosomes. And then after 84 hours, you see just kind of a mass of chromosome fibers completely unseparated from each other. So condensin plays a role not just in cross-linking or holding DNA together, but also in organizing DNA into metaphase chromosomes. Okay, so, and indeed we can take normal chromosomes and either get rid of the condensin with RNA interference techniques or add extra condensin by having them sit in metaphase. We can turn off the cell cycle at metaphase and have them sit in metaphase. And it turns out they load a ton of extra condensin, which you can see in terms of the fluorescence in fluorescence imaging techniques. And what we see is at the same time, the modulus, when we take away the SMC2, goes down. When take away condensin, it goes down. When we add extra condensin, it goes up. And there's a nice correlation between the amount of condensin that's apparently on the chromosomes from fluorescence measurements with the Young's modulus. So indeed, condensin is playing a role of organizing the chromosome, holding it together, and defining what its modulus should be, with defining what the stiffness of that material, of the chromosome material, should be. So, okay, so how do chromosomes get organized and separated from one another in metaphase? Well, so we can get a hint by thinking about what happens when we just pack a lot of strings into a small volume. They're going to get entangled due to the ability of Topo2 to pass DNA through DNA, but how entangled? And it turns out there's a nice scaling law that the entanglement linking number squared. So if you look at the signed linking number of two loops, You pass one loop through the other, that's either +1 or -1, depending on how you define the direction on the fibers, which is defined for chromosomes. So obviously those entanglements could be positive or negative, so their average is going to be 0. But there's going to be a fluctuation, so we can measure the entanglement linking number squared. And that entanglement linking number squared, which tells you basically the number of nearby crossings that are contributing to entanglement, is scaling with the length. Big N here is the length of the whole genome. Little n is the number of chromosomes in the gene that are a number of polymers stuffed into this small volume. And there's a simple scaling law. And what this tells you is that there's two ways to make a genome less entangled. One is to make the chromosomes shorter. If you make them short enough, they'll never be entangled. And in fact, there are genomes which are made up of a lot of small chromosomes which are essentially unentangled. But the other thing you can do is you can change the length of the chromosome by folding it up along its length and reducing the number of persistence lengths. So there's two ways. You can either just make the DNA segments shorter and make big N smaller, or you could actually fold the chromosomes along their length. So that suggests two strategies. So in fact, in large genomes, it appears the chromosomes are folded by folding along their length so that the entanglement linking number squared is going to be tending to go not with the total length of the chromosome, but in fact the length of the chromosome in persistence length units. All right. So how can you do that locally along a chromosome? Well, we introduced the idea some years ago of enzymes that bind, extrude loops for a while. So this is an enzyme, a hypothetical enzyme that binds to DNA, then uses ATP energy, metabolic energy, to pump DNA, pull the DNA in from both sides, pumping out a loop, and then after some time, dissociating. And what happens is the loops grow and reach a characteristic loop size, which depends on how many of those loop extruding enzymes there are around and how fast they extrude loops. And you tend to have formation of a regular loop size. I'll show you how this works in a second. So what's great about this is that this compacts the chromosome into a fiber like we see in cells, but without really ever the possibility of cross-linking, so you don't stick all the chromosomes together. And then the internal tension of these loop domains will tend to drive chromosomes apart. And there's, for biologists, I would describe that there's actually quite a lot of bioinformatic data from so-called high-C experiments, which are quite consistent with this picture. The only problem was back in 2016, actually back in 2012, when we were first talking about this type of mechanism, there were no loop extruding mechanisms, and it was still kind of like science fiction. Since then, it turns out condensin and related SMC complexes are indeed doing exactly this function. This has now been observed in vitro in a variety of ways, and we're pretty sure that SMC complexes do exactly this process of binding to DNA, extruding loops, and then dissociating, and then binding somewhere else. So they are the motorized active loop extruding enzymes. So back in 2016, we, in collaboration with Leonid Murney's group, in particular Anton Golodoborko, did beautiful computer simulations showing how random polymers could be organized into superpolymers of loops by just exactly this mechanism. By binding of loop extruding enzymes, which are shown on the right here as these red blobs along the chromosome, which bind to the gray DNA, or gray chromatin, or gray nucleosome fiber, and extrude it for a while, and then fall off. So they don't do anything else. They're completely stupid. They bind to DNA, they extrude a loop for a while, and then they fall off. And you'll see what happens is you establish a backbone structure without any sequence direction, without any molecular ruler, just the kinetics of these enzymes binding, extruding loops, and then falling off. And you get this nice thick polymer, rather similar to how we see chromosomes being organized inside the cell. Now, because we don't use any in this kind of theoretical study, we didn't use any interactions which are attractive. There's no attractive interaction here. There's just the loop extrusion of the by the loop extruding enzymes. The polymers here, there's two polymers, a blue polymer and a red polymer. They actually try to get away from each other because of osmotic pressure. The excluded volume interactions between the red and blue polymer push the two apart, and you'll see that the red and blue polymer, as the loops are organized along their length, without any condensing interactions, get very dense and push one another apart. Now, in the background acting here is the possibility of passing the red polymer through the blue polymer and red polymer through red polymer and blue polymer through blue polymer. There's effectively topo 2 in these simulations. The polymers are able to pass through one another at some efficiency. So that's very important. You need two things. You need loop extrusion, and you need the possibility of topology change if you're going to disentangle the chromosomes. All right. So turns out that we realized a few years later, actually, my student Sumitava Brahmachari, who's now at Rice University as a postdoc, we realized that if you take a chromosome and organize it with a lot of small loops, it will still be pretty long. On the other hand, if you organize it with just a few really large loops, it'll still be pretty big because it's going to be a big random polymer. So we realized that somewhere in between is a magic loop size, which minimizes the overall length of the chromosome. And here is what the metric that's shown is the amount of entanglement measured as this square of linking number of chromosomes confined within the cell. So because all those chromosomes have to be inside the same cell, they'll tend to get entangled with each other, but you can optimize that entanglement at some characteristic loop size. Well, and it turns out that if you look across the entire range of living things, from bacteria and yeast, actually bacteria have bigger chromosomes than yeast. Budding yeast is the case that I sort of alluded to, which has really small chromosomes. What you see is there's actually a scaling law that describes all of those genomes and their characteristic size, their length inside the cell. And despite the fact that there are quite different types of organisms, and even in the case of bacteria, different type of organization, the optimal loop size model predicts this scaling law, and it actually predicts a particular exponent, 3989, or about 0.44, which describes the experimental data very well. So what we think is that across all of the different organisms, one of the factors which is chosen how cells organize their chromosomes is to optimize the bristle or loop size of these cylindrical brush structures for minimal entanglement of chromosomes with one another. And it turns out we predict a number of other exponents. This was published a couple of years ago in the PNAS. I should just mention that there have been very beautiful observations. This is from Case Decker's lab, published this, I think, in 2018, where a piece of DNA is being studied here in vitro. And Case Decker's group has shown that condensin causes the formation of a loop, which is pulled out by the action of ATP. And they even know something about how the rate depends on tension in that DNA molecule. See if we can run that movie again. Let's see if we can do that. There we go. There's a piece of DNA. Condensin binds here. There's this bright spot. And then that bright spot grows into a loop. Something like what's shown here. And this whole structure is being stretched out with a little bit of hydrodynamic flow, and the DNA is attached to a surface. So, all right. So I'll just mention that rather unbelievably, there was an event in Paris where loop extrusion, the loop extrusion model, was one of the featured physics topics that some musicians, some composers were commissioned, and some musicians were commissioned to play. And I actually attended that concert. It's unlikely I'll ever attend a concert again, which is devoted to playing music inspired by something that I've worked on. In fact, this is one of the pictures from one of our movies. All right, good. So this is what these SMC complexes look like. They're 50 nanometer across structures. And what's really remarkable is that they look pretty much the same in bacteria and in humans, plants, fungus, and other types of so-called eukaryote cells, which are cells with nuclei, despite the fact that the DNA is organized at the sub SMC and loop level quite differently in those organisms. So they all have this loop structure, and there's a certain universality of that complex architecture that goes across all life forms, all living cells on Earth. And in fact, all of the SMC complexes, we've done quite a lot of single molecule study of various protein DNA interactions, including SMC complexes. And what you always see is that these complexes form steps of length along DNA. This is the length of a DNA molecule, watching it go down clunk, 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 clunk with time. This is our own experiments on cohesin from yeast. You can see these steps of about 100 nanometer size. So the step size of these complexes involves capture of DNA loops and a step in the 100 nanometer range, which is about the size that you could conveniently bend DNA or DNA covered with proteins and make a loop. Okay, so here's a picture from one of our recent studies showing these individual loop steps, which is showing the length of a DNA molecule being reduced, clunk, 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 with time after introduction. And this is seen in bacterial SMCs as well as in eukaryote SMCs. So how do we think this actually happens? So the way we think this happens is by an enzyme cycle. This is showing the SMC complex, a kind of a cartoon of it, where you have these long parts of the protein, which are called coiled coil parts of the protein, which are about 50 nanometers long. And there's a lower compartment here. And when ATP binds, the upper arms open. And so you get an upper compartment and a lower compartment, which are topologically separated. They're topologically separated by this chemical bonding of ATP, which joins the two parts of the enzyme together. Then the ATP at some point gets hydrolyzed, it gets burned. And for a short time, you have this upper compartment open and connected topologically. You could move stuff from this upper to lower compartment. So without ATP, you start in this state, you bind ATP, you burn ATP. That's a highly exothermic process. That's a metabolic, a recovery of the free energy stored in ATP. So this is essentially irreversible. And you execute a cycle. So it's very important for any molecular motor that you have a cycle of defined direction. And usually that direction in molecular motors in the cell is defined by use of high energy molecules, which can only, basically are almost impossible to be run backwards. ADP almost never turns into ATP inside the cell except via burning of some other high energy molecule. So now if you imagine you have a DNA molecule trapped in this lower compartment, passing through the lower compartment, when you bind ATP and open the upper compartment, it's possible for that DNA molecule to become grabbed in the upper compartment by, say, an interaction, a favorable energetic interaction with something in the upper compartment, which is now available for binding. Then when the ATP is burned, this bridge goes away, and now this piece of DNA can go through the resulting opening between the upper and lower compartment. Then this thing will close back down to the original state. And what's happened is the arrowhead, which started over here, has ended up over here. So we're coupling the asymmetry of the enzyme in space. There has to be some asymmetry which favors the capture of this DNA loop in this direction, and the asymmetry in time, the irreversibility of the process, to make a molecular motor. So we predicted how this motor should look. It should have a stall force in the low piconewton range, somewhere around half a piconewton, which has been observed actually for condensing. And it should have steps that are in the range of sort of 100 nanometer size steps. So can this really work? So we've recently been doing simulations of condensin moving along DNA using molecular dynamics simulations. And so Stefanos Novidis at the University of Leuven has been doing very nice simulations of this. Let's see if we can run this movie. Oh, is this movie not going to run? Let's see. Let's see if I can start it over. What I'll do I'll leave the slideshow. Oh. I'll leave the slideshow. There we go. And I'll just put this slide up here. I bet that will work. I bet that will work. Okay, let's run that. Now you can see, if I get rid of this stuff over here, a little bigger. There we go. Bigger. Beautiful. Okay, so what you're seeing there is this process realized in a molecular dynamics simulation. So this is with the SMC complex cycling through the three states. And down here, you see the whole DNA molecule SMC complex translocating along the DNA. So translocation has also been observed for these things. And so what's going on is now in this simulation, you can see the complex starts over here. All the complex is doing is changing through these three states and opening. You can see ATP binding, it opens, captures a DNA loop. Why does it capture a DNA loop on a particular side? Because there's a folding of the enzyme that favors capture from one side, so you broken symmetry in space. And then when ATP is burned, that enzyme the enzyme complex moves to the new position along the DNA. So that is how that works. I'll show you this again. So how do you get loops? Well, one simple way to get loops is just to attach the DNA to the bottom of the enzyme. We have some evidence that in yeast, the condensin in yeast does exactly this from other experiments, structural studies. And what happens is the orange loop, now the enzyme complex is translocating along the DNA and pulling the DNA, and it's attached back to itself. So you get a loop that grows with time. So it's a loop growing with time. And then there are a bunch of other schemes, which I won't go into the details of variations on this, that we think explain the phenomenology of how these enzymes work in other organisms. All right, so let's see if we can. Yeah, now I've got this one runs. All right, good. So that's how we think these enzymes work. So I'm going to spend just a couple of minutes talking about cell nuclei. So I've talked mostly about metaphase chromosomes, which are those beautiful separated chromosomes which appear inside our cells when they're going through cell division. But most of the time, in many of our cells, almost it could be many years, cells spend with a nucleus sitting inside the cell. And so we were interested in the mechanics of the nucleus and how the cell controls a nice kind of round nucleus and how DNA plays a role in controlling that. And so postdoc of mine, Andrew Stevens, who now has his own faculty position in University of Massachusetts in Amherst, developed techniques for isolating whole nuclei. So you can see this nucleus being pulled out of the cell. There's the rest of the cell over here off to the side. Takes the cell, takes out the nucleus, and then you can do mechanical experiments on a whole nucleus that has all the chromosomes inside the nucleus and a completely different organization from the metaphase chromosome I was talking about. Okay, and people before had done mechanical studies on nuclei, but typically by sucking the nucleus and maybe the cell around it into a pipette and using it kind of like a piston to do measurements of the mechanics. And it turns out to be rather complicated, and you tend to see a lot of flow effects. You can imagine there's a lot of flow in that kind of experiment where you're sucking. We wanted to do static mechanical measurements, kind of like we've done for metaphase chromosomes, to really look at the static mechanics. So Andrew developed this technique, just like for the metaphase chromosomes, where he could stretch a whole nucleus. Okay, so what he found was the whole nucleus is quite stiff and has a stiffness, a human nucleus, that takes a nanonewton to even add 1 or 2 microns to its length. And the size of a nucleus is about 10 microns across for a human nucleus from a human cell. And so this is a range of about 2 micron stretching or about 20%. Then he observed that around 20%, 25%, you saw a change in slope of the force response. So it looks like there's 2 things going on: short force response and then a long stretching response. And we saw this in a variety of types of cells. We could quantify this quite precisely. Well, what we can do is we can do the same game as we played with metaphase chromosomes. We can get rid of the DNA by cutting it up. And so what we found was when we took original nucleus that has this initial force response of to about a 20% change in length, and then we treat it with an enzyme called micrococcal nuclease. It cuts between all the nucleosomes, basically completely cuts up the DNA. What happens is the nucleus becomes really hard to see, and the stiffness goes completely away for that initial stiffness. But the secondary stiffness from stretching just the boundary of the nucleus remains. And so we are able to separate the stiffness of the interior of the nucleus from the stiffness of the boundary. And this actually was quite unexpected, that so much of the elasticity of the nucleus actually comes from chromatin itself. Well, so now I can, and it turns out we can recapitulate this in the simulations as well. Ed Bannigan did very nice molecular dynamics simulations where he showed that indeed you could get this bimodal elasticity of the initial elasticity being provided by the bulk here, and the secondary elasticity being provided by the surface by simply defining different strength springs for the surface region and the interior region. And he was able to do the same kinds of numerical experiments that we had done real experiments. All right. So it turns out that there are many situations, usually pathological, where the nucleus can rupture. And that's very bad, because if stuff from the cytoplasm gets into the nucleus, stuff from the nucleus gets into the cytoplasm, usually it's a bad thing for the DNA inside the nucleus, which can cause genetic damage and has bad consequences for the future life of that cell, maybe even for the whole organism. So we were interested in how the cell regulates the whole nucleus and its topology, whether there are holes or not and it's bursting, and its mechanics and its geometry. Well, so the cell has a mechanism to do that. It turns out that the nucleosomes in a human cell and all types of eukaryote cells, they can have chemical labels on them that the cell can change dynamically in time. So now I'm introducing the idea to you that the chromosomes inside the nucleus at interphase actually have along them kind of 2, you can think about, I'm just going to talk about 2 flavors of paint or 2 colors of paint. So they can be more active. We actually call that A-type regions of the chromosome, or euchromatic is another term that's used, where they're highly acetylated. The nucleosomes have a lot of acetyl groups on them at defined positions. And the machinery inside the cell interprets those acetyl groups as, oh, this is active genetic material, and kind of unfolds it and it tries to interpret it as genetic information for making messenger RNA. On the other hand, those same sites, which can carry acetyl groups, which can drive euchromatin, can also be methylated, just as methyl group can be added. And those methyl groups are interpreted as, oh, this is not material in the chromosome which is meant to be genetic information. Don't turn this into messenger RNA and in fact fold it up into a tightly organized heterochromatic domain. And those are often called B compartments, B regions of the chromosome or heterochromatin. Well, it turns out we can manipulate the cell to make more heterochromatin or less heterochromatin. One way to do that is drugs. We can treat the cells with drugs, and an example of a drug is VPA, which biases the chromatin towards being more euchromatic. And indeed, we can see that the stiffness of those nuclei goes down because the chromosome is more unfolded and the nuclei are softer. At the same time, we can drive the methyl groups up in quantity using other types of drugs, in particular something called methylstat, which tends to cause methyl groups to accumulate on the nucleosomes, and the chromosomes, the nuclei, get stiffer. And there are a whole bunch of drugs that we've used, which are commonly used. These are called epigenetic drugs because this is information stored on chromosomes, which is not genetic. It's epigenetic. It's metagenetic, if you like. It's not associated with the DNA base pair sequence, but nevertheless is genetic information on the chromosome. So there's this second level of epigenetic coding going on. All right, and I'll just mention that what we can do is if we take heterochromatin away with drugs, and these are experiments with a particular drug called DZNEP or with the drugs I mentioned in the previous slides, we indeed see more blebs and ruptures. In fact, here's a movie of a cell nucleus that is rupturing, and this green stuff is a protein that's mobile inside the nucleus. Play that. There it goes. Well, as we see the movie progress, boom, the nucleus ruptures and the green stuff goes to the outside of the nucleus. So this is a nice tool to monitor. These blebs, which eventually lead to ruptures, tend to be along the major axis of the nucleus in the region of highest curvature. So it might actually be that the blebs tend to form in the region of highest curvature because of mechanical stress. And so that's still a topic of quite a lot of so we have also done experiments on, that's the last thing I'll mention, the protein that I mentioned right at the beginning called HP1 alpha that cross-links methyl groups together. So now I'll reveal what HP1 alpha does is it links two methyl groups together on nucleosomes, thus forming condensed heterochromatin. And indeed, in cells where there's new technology now where you can take cells that have been slightly genetically modified so that a particular protein, in this case HP1 alpha, can be made to disappear in about an hour. So there's a particular way of setting up an experiment where you add a drug and bang, all of the protein of a particular species in the cell goes away. And those cells, before you add the drug, have nice round nuclei. After you add the drug, they have these terribly shaped nuclei, and their spring constant goes down. And in fact, the nuclei start to rupture more often. So what's very important inside the cell, between cell divisions, is actually to use mechanisms like HP1 alpha cross-linking to define the shape of the chromosome, the shape of the nucleus, and to protect it against rupturing and having these kind of funny shapes. Okay, so the mechanics of the nucleus are very dependent on heterochromatic cross-linking mechanisms, and so that's been a subject that's been quite interesting to study. So our picture is that normal cells, well, they can get into trouble, and this does happen in some diseases, where there's too much euchromatin or too little heterochromatin, and then they have a weak nucleus that can easily bleb or rupture. And it's possible to imagine taking a cell that has a deficiency in nuclear nuclear stiffness or permeability, and to add heterochromatin to it to rescue its tendency to make blebs or ruptures, or peculiarly shaped nuclei can perhaps be restored to normal shape. And so Andrew at his new lab is working on really fleshing out how external cues, signals coming to the cell, we already know about a whole class of these signals whereby tension can be sensed in the cell, which can change the amount of hetero and euchromatin, therefore change the shape of the nucleus and control the amount of DNA damage inside the nucleus. And so actually, Andrew has actually demonstrated that using stress sensing ion channels at the cell surface. Okay, so that's what I wanted to tell you guys about today, is that chromosomes have to be geometrically and topologically self-organized. A key mechanism is active loop extrusion by SMC complexes, which can help to disentangle chromosomes by telling Topo2, the disentanglement enzyme, how to disentangle DNA. Metaphase chromosomes are not really condensed. They're still under pretty good solvent conditions and are not supported by a protein scaffold. They're supported by packed together loops. And I've told you a little bit about cell nuclei. In particular, most of our experiments have been done in humans, or a few in mice, where we see that definitely the mechanics and stability of the cell nucleus is controlled in part by chromatin and by the epigenetic marks that are along chromatin, in particular the heterochromatin, euchromatin balance. Okay, and this work has been a result of a huge number of collaborations and cooperation of lots of people at different institutions. I've talked about a few of them during the talk. And thank you for listening. I'd be happy to take any questions you have.
Yeah, thank you, sir, for your wonderful presentation. So we have few questions. So if you allow, we can start our discussion session, sir.
Yeah, sure.
Okay, so the first question, what is the nuclear region?
I didn't hear. Say again.
Yeah, so what is the nuclear region?
The nuclear region of the cell. So first of all, you have all of the chromosomes in the interior. And the chromosomes are organized by a lot of proteins that are binding together. And then the boundary of the nucleus is actually a very well-defined structure. There's 2 layers of cell membrane. And then there are proteins that are cross-linked like a surface layer, like in Ed Bannigan's simulation I showed you. That's actually meant to be a model for cross-linked proteins called lamins, which form a kind of a 2-dimensional mesh on the interior of the nuclear envelope. So that nuclear envelope, it's not just plasma, it's not just lipid bilayer, it's lipid bilayer with polymerized protein, really defining that round shape. And then on top of that is all of the chromosomal material, the heterochromatin, which gives the nucleus its mechanical stiffness and its shape. So that's all. Those are the sort of key components of the nucleus that I've talked about for this talk. Of course, the nucleus contains a gazillion other structures as well to carry out specific biological functions.
Okay, sir, thank you. We have another question. So what is cohesin?
Cohesin. Yeah, I didn't talk too much about cohesin.
Yes, yes.
So, but it's extremely interesting enzyme complex. So you remember at the beginning, let's go back to the, on. Huh. Is it really dead? Oh, a little slow there. So cohesin, so okay, in our cells, human
Yep.
cells, it turns out that we have 3 basic types of SMC complexes. And this is true in most eukaryote cells. We have condensin. Condensin is involved in folding chromosomes up during cell division, and it's beginning to appear it plays some role in defining loop organization between cell divisions as well. And its main function seems to be to make sure chromosomes are not entangled with each other, okay? That's condensin. And so I showed you, simulations of how we think condensin works and how it can organize. chromosomes into loops that will push each other apart, and you can disentangle chromosomes out. So cohesin, on the other hand, plays two roles. One is it plays some role. Actually, the original function that was discovered by biologists was that it holds the two sister replicated chromatids, as they're called, the two chromosomes that are eventually going to be pulled to get apart during cell division. It holds those two replicated sister chromosomes together until the right moment when they can be released. But it turns out that it has a crucial role during interphase as well, when genes are being expressed, when messenger RNAs are being made by transcription. And what cohesin does is it's responsible for folding the genome into structures, into loops, which are defined by DNA sequence. Now, this is still not entirely understood how this works, but it involves this protein CTCF that binds at the boundaries of many of these loop structures. And cohesin seems to extrude loops, just like, I was describing, it's a loop extruding enzyme, but it stops when it hits CTCF. And so this is a nice way for the cell to regulate particular sequence-defined loops between cell divisions. Condensin, as far as we know, does nothing with DNA sequence. It has no dependence on DNA sequence. But cohesin does, and during interphase, it can define DNA loops.
Okay, sir.
I was just going to mention that
Okay.
cohesin has this other function, and it turns out there are a few different. Cohesin is a little smarter than condensin in some sense, in that it has some sensitivity to transcription factors and plays some role in folding sequence-defined loops. I just wanted to mention that it's been a mystery for decades how large loops in the genome that are sequence defined were actually being folded. And now we more or less know it's due to loop extrusion by cohesin. So that's been a major development in the last 10 years. I'll just mention that there's a third SMC complex called SMC 5,6, and we don't really understand what that does, but it probably has some role in repair of DNA. You can imagine if you're trying to repair breaks in DNA inside the cell, it could be a good idea to be able to extrude some loops to figure out where the breaks are. And so it seems to be have some role in repair of DNA breaks.
Yeah, thank you, sir. This may be the last question.
Oh,
So,
yeah,
what are
sure.
the difference between Eurochromatin and heterochromatin?
Yeah, good. Okay, I went through that all very quickly. What I was going to do. I went through that very quickly. I'll put up this slide again. So, euchromatin. So basically, inside our chromosomes, so the protein DNA complex. You guys can still hear me, right? The protein DNA complex, remember, is a nucleosome, and our chromosomes are made up into long DNA molecules formed into nucleosomes, a sequence of nucleosomes. Actually, it's almost packed together along the chromosome. Those nucleosomes are large protein structures with a lot of amino acids. So it turns out that each nucleosome contains very well-defined proteins, a total of 8 proteins. Each one is about 100 amino acids long, so you got 800 amino acids in each nucleosome. And euchromatin is typically attributed to nucleosomes which are carrying particular histone marks which favor transcription of genes. Now, for those who are experts, one of the canonical euchromatic marks is histone H3, lysine number, the amino acid residue number 9 happens to be a lysine, so we call that H3K9, and it has an acetyl group at that point added to that amino acid. However, if H3K9 is methylated, it turns out that it will act as heterochromatin. So those chemical marks are binding sites for other proteins, which will either recruit RNA polymerase, which will make messenger RNA and tend to unfold that region of the genome, or they can recruit other proteins like HP1 alpha in the case of H3K9 dimethyl trimethyl, which stick together the nucleosomes into a mass that is quite stiff and contributes to nuclear stiffness, but also is very hard to transcribe, very hard for the enzymes to get in and transcribe. So it's an extremely good example, a heterochromatin euchromatin switch is an excellent example of how a chemical change can trigger a physical change of the arrangement of nucleosomes, which can then control genetic processes like transcription. Basically, in heterochromatin, the machinery of transcription just can't get in there because the heterochromatin is folded up by crosslinking by HP1 alpha. So heterochromatin actually was the name comes from the fact that inside the cell, by microscopy, you can see dark regions of the nucleus. I'll show you a picture. You know, if we look at this original picture I showed you, so you can see that there's this dark stuff here. That's heterochromatin arranged against the nuclear envelope. That's the stuff that actually is making the nucleus stiff. You might wonder what these big blobs are. These are structures called nucleoli, and they're the regions where ribosomes are being made. And they're very interesting structures. They're sort of neither heterochromatin nor euchromatin. And you can see these blobs of dark stuff in the nucleus. Those are heterochromatic regions, condensed regions of the nucleus. And then there's lighter regions of the nucleus. Those are the euchromatic regions. So in fact, the heterochromatin, the word means different chromatin. It's the dark stuff. The euchromatin is the light stuff. But we now know that they correspond to regions of the DNA and the chromosomes, which are carrying nucleosomes with specific chemical marks. So the modern way of thinking about heterochromatin, and of course we have different classifications of different kinds of heterochromatin as well, which I won't go into right now, is in terms of chemical identity. So they're actually, we now understand the chemical marks which are present. We can even manipulate them to play games with what the cell is doing with its chromosomes. I hope that's helpful.
Yeah, thank you, sir, for your wonderful presentation and discussion session. As we don't have any more questions, so we can conclude our discussion session.
Okay.
So thanks again for joining with us, and
oh no,
it was
my
a great
pleasure.
webinar. Yeah, and the main aim of our program is to motivate, and I think our students have motivated by your lecture. So thanks again for accepting our invitation, sir, and hopefully in the near future we can arrange another webinar with you. Bye for today.
All right.
Have a nice day.
All right, very good.